Hello, I'm Yan Liu from Professor Chang Huiyang's group at Caltech. Today I will describe our efforts to achieve fluorescence imaging through dynamic scatter media such as living biological tissue. Fluorescence imaging is indispensable to biomedical research. It is one of the main tools we use to study life. On the left, we observe cell division. On the right, we observe neural activity in the brain of a larval zebrafish. One of the reasons why larval zebrafishes are so widely used in biology is because they are transparent, so scientists can do fluorescence imaging at high spatial resolution. However, while larval zebrafishes are transparent, as seen from the left, adult zebrafishes are opaque, as seen from the right. The opaque nature of adult zebrafishes prevents fluorescence imaging at high spatial resolution. This is a common problem when we image through scatter media such as milk or biological tissue. Light is scattered due to the refractive index heterogeneities within this media. As a result, we cannot use a lens to focus light or image an object deep inside the scatter media. For coherent light, the light intensity distribution inside this media looks like this, which we call a speckle pattern. In general, light scattering prevents optical focusing beyond 1 mm deep inside biological tissue, which we call the optical diffusion limit. One way to achieve fluorescence imaging through scatter media is by speckle correlation imaging. It uses the fact that the autocorrelation of a speckle pattern is a sharply peaked function. However, these methods rely on the memory effect, so they work for relatively thin scattering samples. Another way to achieve fluorescence imaging through scatter media is by feedback-based wavefront shaping and scanning the focus with the memory effect. Light is scattered in a scatter medium, and a speckle pattern forms because of the random phasor sum of the electric field associated with each beamlet. By adjusting the phase of each beamlet to maximize a feedback signal, people can make the phasors aligned and achieve a focus inside scatter media. This is called feedback-based wavefront shaping. By using linear or nonlinear fluorescence signals as the feedback signal, people can focus light on a fluorescent particle. Then they can scan the focus with the memory effect and perform fluorescence imaging. Again, the focus scanning relies on the memory effect, so it works only for thin samples. We need a way to raster scan the focus without using the memory effect. One way to do this is to use ultrasound. Ultrasound is much less scattered in tissue due to its long wavelengths, and we can raster scan the ultrasound focus easily. Here, my colleagues use ultrasound modulated light as a feedback signal in wavefront shaping. By maximizing the feedback signal, they can focus light at the ultrasound focus. Then, it is possible to raster scan the optical focus by scanning the ultrasound focus and doing wavefront shaping at each ultrasound focus location. Then, it is possible to do fluorescence imaging with this raster scanned focus. However, one thing we should note is that the feedback-based wavefront shaping involves repetitively generating a wavefront and measuring its associated feedback signal, so it is relatively slow. For example, in their experiment, it takes several hours to achieve a focus. This low speed is a major problem for achieving a focus within living biological tissue because speckle decorrelates much faster in living tissue compared with the runtime of feedback-based wavefront shaping. Living biological tissue is not only a scatter medium, but also a dynamic scatter medium, where all these light scatters keep moving due to physiological motions such as breathing, heartbeat, and blood flow. Red blood cells are the main fast-moving scatters. If photons are singly scattered by blood, the speckle correlation time tau c, which quantifies how fast speckle pattern changes, can be estimated by lambda over flow speed, which is on the order of a millisecond. The speckle correlation time is even shorter if photons are multiply scattered by blood. This video shows the speckle patterns of light passing through a living mouse ear before and after blocking blood flow. Indeed, light scattered by red blood cells causes speckles to change with a speckle correlation time on the order of a millisecond, as can be seen from this figure. When we block the blood flow, the speckle correlation time was much longer. Therefore, to achieve a focus in vivo, we should complete wavefront shaping within 1 millisecond before speckle decorrelates. We also measure the speckle correlation time in the living mouse brain. We insert a fiber into the brain to create a point source and then measure the speckle patterns outside the brain. The result shows that the speckle correlation time decreases with increasing depths, and it becomes shorter than 1 millisecond at a depth of around 2 millimeters. Because feedback-based wavefront shaping methods generally takes much longer than a millisecond to achieve a focus, we need to improve its speed or use some other methods for in vivo applications. The method we have been working on for focusing light inside scattering media is called guide star assisted optical phase conjugation. This method is much faster than feedback-based wavefront shaping because we can determine the optimum wavefront globally in a full-field manner without doing any iteration. In this method, a guide star emits light inside the scatter medium, and we detect the scattered light field outside the medium. Imagine if we can do time reversal, the detected photons will trace their trajectories back to the guide star where they come from. In this way, they form a focus despite scattering. However, in reality, we cannot reverse time. Fortunately, we can do something called optical phase conjugation using this phase conjugate mirror to achieve the equivalent time reversal. By definition, optical phase conjugation of a light field E0 is achieved by conjugating the phase of E0 at each frequency. Mathematically, we can prove that phase conjugation is equivalent to time reversal. Here is a comparison between a regular mirror and a phase conjugate mirror that performs optical phase conjugation. A regular mirror would reflect a beam to this direction, while a phase conjugate mirror would reflect the beam back to where it comes from, as if time has been reversed. So far, our labs have developed several guide stars for focusing light inside scatter media with optical phase conjugation. These guide stars include focus ultrasound, dynamic permittivity, magnetic particles, and photoswitchable proteins. Because focus ultrasound is non-invasive and easily controllable, I will mainly talk about works with the ultrasound guide star. Ultrasound-guided optical phase conjugation is also called time-reversed ultrasonically encoded optical focusing or true focusing. First, we focus ultrasound deep inside the scatter medium. Then, we illuminate a laser beam. Of course, light will be scattered by all these heterogeneities, but a portion of light passing through the ultrasound focus will change its frequency due to the momentum transferred from the ultrasound. These frequency-shifted photons shown in red come from the ultrasound focus, and we call them ultrasound-tagged photons. We detect the ultrasound-tagged photons and do an equivalent time reversal by phase conjugation. Then, these photons will trace their trajectories back to the ultrasound focus where they come from, thereby forming a focus. True focusing has been applied by my colleagues to achieve fluorescence imaging through scattering tissue. They use true focusing to generate a focal spot inside scattering tissue to excite fluorophores. Similar to other point scanning microscopy, by recording the fluorescent signal as a function of true focusing position, they can acquire an image of the object. This fluorescent object was embedded in a piece of chicken tissue 5 mm thick. Because of scattering, we cannot see the object with conventional fluorescence imaging. However, with true focusing, we can see the object clearly. You notice that this experiment was done in ex vivo tissue because it takes 4 seconds to achieve a focus. To apply true focusing to dynamic scatter media such as living biological tissue, we need to overcome the speckle decorrelation problem and improve the true focusing speed. Next, I will introduce our efforts. Before that, we need to first understand how to achieve optical phase conjugation and true focusing in reality. There are two approaches. In one of the analog approaches, we use a reference beam tuned to the same frequency as that of the ultrasound tag light. They interfere and write a hologram inside a photoreflective crystal, in which the refractive index distribution follows the light intensity distribution in the interference pattern. Then, we use another reference beam to illuminate the crystal, and the diffracted light would be phase conjugate to the ultrasound tag light, so the diffracted light would converge to the ultrasound focus. Based on this principle, we developed high-speed true focusing using a fast-responding photorefractive crystal called SPS crystal. The SPS crystal is made of tellurium-doped tensile hyperdiphosphate. During experiments, we moved the scatter media to create time-varying speckle patterns like this to mimic the dynamic nature of speckles within living tissue. Even when the speckle correlation time was 5.6 milliseconds, we can still achieve true focusing. We embedded an absorptive object in between the two scatter media. By scanning the true focus across the object, and measuring the transmitted light signal, we can obtain an image of the object. Even when the scatter medium was changed to a living mouse here, we can still obtain an image of the object. This is the first demonstration of true focusing within living biological tissue. I just talked about how we achieved high-speed true focusing using a fast-responding photorefractive crystal. The drawback of this approach is that the reflectivity of the phase conjugate mirror is low, meaning the time-reversed light is much weaker compared with the ultrasound tag light. To achieve a higher reflectivity, we switch to an optoelectronic approach. This approach achieves optical phase conjugation by following its definition. First, we measure the wavefront of the ultrasound tag light using digital holography. Then, we display the conjugate wavefront on a spatial modulator that modulates the phase of light. Upon reflection, a planar reference beam acquires this phase and becomes phase conjugate to the ultrasound tag light, so the modulated light would converge to the ultrasound focus. As you can see, this approach involves two steps. One is wavefront measurement, the other is wavefront generation. However, both steps are slow. First, I will introduce how we improve the wavefront measurement. We use a lock-in camera that can measure the wavefront within 0.3 milliseconds. As a test, these are the standard wavefront we measured. Other than speed, there are some other advantages of using this camera for true focusing, which I don't have time to talk about. Here is the wavefront of the ultrasound tag light we measured within 0.3 milliseconds. By displaying the conjugate of this wavefront on a spatial modulator, we focus light inside the scatter medium. Having talked about how we improve the speed of wavefront measurement, let me talk about how we improve the speed of wavefront generation. Here is a comparison of different spatial modulators used for wavefront modulation. Pneumatic liquid crystal-based spatial modulators are the most popular. However, they usually take tens of milliseconds to modulate the phase, which is much longer than the 1 millisecond requirement for achieving true focusing in vivo. Digital micromirror devices can be faster. However, they normally achieve binary amplitude modulation, which results in low focusing efficiency and contrast. In our work, we use ferroelectric liquid crystals as a fast spatial light modulator. The spatial light modulator has the shortest runtime of 1 millisecond, including data transfer time. However, unlike pneumatic liquid crystals that can do full phase modulation, ferroelectric liquid crystals can do only binary phase modulation. Fortunately, we proved that this binary phase modulation scheme only reduced the focusing contrast by around half. However, we gain more than 10 times in speed. Here are the physical pictures of the above three devices for wavefront shaping. Now that we use an SLM that achieves binary phase modulation, we don't need to measure the wavefront very accurately. We develop a binary wavefront measurement method that works perfectly with binary phase modulation while doubling the wavefront measurement speed. This is a representative binary phase map we use to correct for scattering. With this improved speed, we focus light through 3 mm thick dynamic chicken tissue, even when the speckle correlation time was 2 milliseconds. We also focus light inside dynamic chicken tissue, even when the speckle correlation time was 5 milliseconds. True focusing is great because it enables fluorescence imaging with scattered light, so we can image deeper. However, although I built the fastest true focusing system in the world, the speed is still not high enough to tolerate the fast speckle decorrelation that occurs in living tissue. Can we address this challenge? We developed a technique called flux, which stands for fluorescence and ultrasound modulated light correlation. Instead of fighting with the fast speckle decorrelation, we instead use speckle decorrelation to achieve fluorescence imaging through dynamic scatter media such as living tissue. Let me explain the principle of flux. Suppose we have some fluorescent beads that we want to image inside this dynamic scatter medium. We probe the sample with a pulse laser beam. Similar as true focusing, we apply ultrasound to tag the photons that pass through the ultrasound focus, and we detect the ultrasound tag light with this camera. Simultaneously, we detect the fluorescent light emitted from the object using this photodetector. Then, we calculate the correlation coefficient of the two signal traces. To explain why we do this, let us consider two cases. In the first case, the ultrasound transducer is at position 1, so that the ultrasound focus overlaps with the fluorescent bead, as shown here. In the second case, we move the transducer to position 2, so the ultrasound focus is away from the fluorescent bead, as shown here. Because this is a dynamic scattering medium, such as living tissue, the light intensity at each location inside the medium keeps fluctuating, and different locations have different fluctuations. In the first case, because the ultrasound focus overlaps with the fluorescent bead, they experience the same fluctuation of the excitation light. Therefore, the fluorescent light and the ultrasound tag light fluctuate in a similar way, and their correlation coefficient of the two signal traces would be high, close to 1. However, in the second case, because the ultrasound focus is away from the fluorescent bead, they experience excitation light with different fluctuations. Therefore, the fluorescent light and ultrasound tag light fluctuate in a different manner, and their correlation coefficient is low, close to 0. Based on this observation, we can obtain an image of the fluorescent object by plotting the correlation coefficient of the two signals as a function of the ultrasound focus position. Let me show you some experimental results. Here we have two fluorescent beads. When we put them inside the dynamic scattering medium, because of strong scattering, we cannot identify the beads under a conventional fluorescent microscope. However, with flux, we can image the beads inside the scattering medium. In this image, each pixel value is not light intensity, but the correlation coefficient of the two signal traces. For example, at this location where the ultrasound focus overlaps with the fluorescent bead, the ultrasound tag light and the fluorescent light fluctuate in a similar manner, and their correlation coefficient is high. However, at this position where the ultrasound focus is away from the fluorescent bead, the two signal traces fluctuate in a different manner, and they have a low correlation. This image is an interpolated version of the image on the left to remove the pixelation effect. Again, we can see the two fluorescent beads clearly. This result was achieved when the speckle size was 39 micron, close to the ultrasound focal size. In practical scenarios, the speckle size can be smaller, so there can be multiple speckle grains within the ultrasound focus and the fluorescent object. We analyzed the signal-to-noise ratio in this case in the next slide. Here, A denotes the fluorescent object shown in pink, and B denotes the ultrasound focus shown in blue. Their overlapped region is denoted by X. Each small circle is a speckle grain. NA, NB, and NX denote the number of speckle grains within each region. For simplicity, in the following analysis, we assume that the fluorophore concentration is uniform within the fluorescent objects. For the more general case, please see our supplementary note 1. When the number of detected speckles formed by ultrasound tag light is much larger than the number of speckles within the ultrasound focus, which is usually the case, the SNR can be written as this, where ND is the ratio between the total measurement time and the speckle correlation time. Mu is the power per speckle. Alpha and beta are constant factors related to the detection efficiencies of the two signals. When the power per speckle and the detection efficiencies are sufficiently large, the SNR can be simplified to this equation. When the speckle size decreases, NA, NB, and NX increase by the same factor. Therefore, the expression NX over square root of NA, NB remains a constant, and the SNR does not change. Here, we show that when there are around 200 speckle grains within the ultrasound focus and fluorescent object, we can still image the object through a dynamic scattering medium. As a control, the top right figure shows that if we don't use flux, we cannot see the object through the scattering medium. We can compare true and flux for fluorescence imaging. In true, we need to first measure the wavefront of the ultrasound tag light using holography. A camera records multiple frames of interferograms, transfer the data to a computer where we reconstruct the phase map. Then, we display the conjugate phase map on a spatial light modulator, which generates time-reversed light that converge to the ultrasound focus and excite the fluorophores. For in vivo brain applications, the whole process has to be completed within 1 millisecond before speckle decorrelates. However, with current technology, data transfer and phase modulation by an SLM alone takes at least 1 millisecond. In flux, we record the fluorescent light and ultrasound tag light as a function of time. For in vivo applications, we just need to make sure the measurement time for each data point in the signal trace is shorter than the speckle correlation time of living tissue. This is easier to achieve because ultrasound modulated light can be measured within a microsecond in theory, while the speckle correlation time is 1 millisecond. In flux, data transfer and processing can be done offline, while in true, we have to complete this whole process on the left in real time. Therefore, compared with true, flux can tolerate speckle decorrelation 3 orders of magnitude faster. In summary, light scattering prevents optical focusing and imaging beyond 1 mm deep inside biological tissue. To break this optical diffusion limit, we developed guide star-assisted optical phase conjugation techniques such as true and trap focusing. For in vivo applications, we need to improve the speed of the system to be shorter than the speckle correlation time. To achieve this goal, we developed high-speed true focusing based on an SPS crystal, a lock-in camera, a ferroelectric liquid crystal-based spatial light modulator, and a binary wavefront measurement method. We also used the lock-in camera to increase the bit efficiency and SNR during wavefront measurement in true focusing. We invented flux for fluorescence imaging through dynamic scatter media. Instead of fighting with the fast speckle decorrelation, flux uses speckle decorrelation as a coding mechanism to perform imaging. In theory, flux can tolerate speckle decorrelation 3 orders of magnitude faster than true focusing. I'd like to thank my advisors, co-authors, lab members, and the funding from NIH. Thank you for your attention.